Good day to all of you. Today I'm going to present before you on the topic Impurity Profiling and Degradation Study of Drug Substances and Drug Products. The factors determining safety of drug products. First is pharmacological and toxicological profile of the drug substance, the relationship of beneficial and adverse effects of the drug, because the toxicological profile of the drug will determine the safety and efficacy and adverse effect of the drug. The impact of impurity present in drug substance. The analytical chemists play an important role in monitoring and controlling the impurities in drug substances and drug products because they are the one who carry out the stress testing studies and all the degradation impurities or toxic chemicals that arise as a result of degradation studies or also which is present in the raw materials or during the synthesis and manufacture of the drug substance and drug products or during the stability study. All of them are characterized by the analytical chemist. They are isolated and also accordingly they are characterized and identified and they are reported, identified or qualified by the pharmacologist later on. The guidelines governing the impurity levels in drug substance and drug products, ICH Guidelines Q3A, lays down the impurity testings, impurity in drug substance. Any component of the drug substance that is not the chemical entity defined as the drug substance is an impurity. ICH Q3B elaborates the impurity testing in drug products and the specification of the drug products, what should be the limits for reporting, identification threshold, and also qualification threshold for all the degradation impurities arising as a result of manufacture or stability studies. ICHQ3C elaborates the limits of residual solvents uh, that would, if present within the drug product, would impact the efficacy of and toxicity profile of the chemical new drug entity. There are different types of impurities. What is an impurity? First of all, a molecule resulting from a change. This is an ICH definition for an impurity. A molecule resulting from a change in the drug substance brought about over time. For stability testing of the products, such changes could occur because storage or processing, for example, oxidation, deamination, proteolysis, and aggregation may cause impurities. There are different types of impurities, organic impurities, inorganic impurities, and residual solvents. Organic impurities can be formed during manufacture process or during the storage of new drug substances. That is, they can be the starting materials which are of less purity, intermediates are of less purity, byproducts, drug products, reagents, ligands, and catalysts. So all of them are present, may be present as organic impurities in the drug substances. Inorganic impurities, they can arise during manufacturing process. Normally they are known and identified. Example, reagents, ligands, catalysts, heavy metals, other residual metals, inorganic salts and other materials like filtrates and charcoal, which is all they are the potential source of impurities in the drug product because mm, during the manufacture, excipients used in the manufacture, or even for that matter, if the API is of less purity and contains all impurities of their starting material or solvents, the drug substance can also have impurities. Apart from that, the excipients may also have impurities that could be either heavy metals or residual solvents formed during their preparation or manufacture. So all these are impurities. Residual solvents may be present at the time of manufacture. Some residual solvents, they're not dried adequately, so they can remain in the drug substance and they are impurities and they are considered as impurities in the drug substance. They are generally inorganic organic liquids that are used during synthesis of drug substances as a vehicle for preparation of solution or suspension. ICHQ3C guideline provides the limits of residual solvents based on existing safety and toxicity data. They were classified in three categories: Class 1, Class 2, and Class 3. Class 1 is the most toxic and environmentally hazardous solvents that needs to be avoided completely. Class 2 considered a lesser risk that may be considered for use but within certain limits. It should be present. It should not be present beyond certain limits in the finished product. And Class 3 are lowest risk category. Their limits also have been elaborated in the ICH guidelines. Accordingly, the specification of the finished products should be prepared. So now identification and qualification threshold of impurities in drug substances and drug products have been elaborated as per by the ICH guidelines. So if the maximum daily dose is less than or equal to 2 gram for an API drug substance, the reporting threshold is 0.05%. Identification threshold is 0.1% or 1 mg per day, whichever is lower. And the qualification, that means qualification identification is identification of the chemical structure. So if it is beyond 0.05%, they need to be identified. If it is if it is beyond 0.1%, the chemical structure needs to be identified. And if it is beyond 0.15%, the toxicity data should be available. For finished product, if the maximum daily, if for the drug substance, if the dose is maximum daily dose exceeds 2 gram, then the reporting threshold is 0.03%. That means if any impurity observed, unknown impurity observed beyond 0.3%, they need to be reported. If observed beyond 0.5%, they need to be identified for the chemical structure. And if it is observed beyond 0.05%, they also need to be qualified. For drug product, if the maximum daily dose is less than 1 mg, it's 1% of 5 microgram total daily intake, whichever is lower. If it is beyond 0.15% or 1 mg per day, whichever is lower, the qualification threshold has to be. That's a qualification threshold and it needs to be evaluated for toxicological study. If the dose is between 1 to 10 mg, the reporting threshold is 0.5% or 20 mg microgram of the total daily intake, whichever is lower, and the qualification threshold is 0.05%. If the daily dose is between 10 mg to 100 mg, there are no reporting threshold or identification threshold. However, the qualification threshold is 0.5% or 200 microgram of the total daily intake, whichever is lower. If the dose is greater than 10 mg, again the qualification threshold is 1% or 50 microgram of the total daily intake, whichever is lower. If the dose is greater than 10 mg and but less than 2 gram, 0.2% or 2 mg of the total daily intake, if it is any impurities beyond observed beyond 0.2% or 2 mg, then they need to be reported as well as identified for the chemical structure. If it is greater than 100 mg but less than 2 gram, 0.2%, then if any impurity is observed it's observed beyond 0.2% or 3 mg total daily intake, they need to be qualified. If it is greater than 1 gram and an impurity observed beyond 0.1% should be reported. If it is less than 1 gram, any impurity observed beyond 0.1% should be reported. And if it is greater than 1 gram, any unknown impurity observed beyond 0.05% should be reported. If it is greater than, if the total daily intake is greater than 2 gram, maximum daily dose of the drug is greater than 2 gram, then any impurity observed, unknown impurity observed beyond 0.1% should be reported and identified. And if it is observed beyond any unknown impurity beyond 0.15% should be qualified. So these are details of the various guidelines which has been published by the various regulatory agencies throughout the world, international agencies, ICH. These are the different guidelines. related to different impurities. EMA, these are the different guidelines related to different impurities. US FDA has elaborated guidelines for various genotoxic and carcinogenic impurities. TGA Canada have laid down the guidelines for impurities in existing drug substances and products that were published on 6 September 2005, and TPD Australia uh, have published guidelines for Australian regulatory guidelines for prescription medicines, Appendix 1.8, impurities in active pharmaceutical ingredients and finished product on June 2004. So now there are various sources of impurities which we already discussed before. It could be either synthesis related, the raw materials which are used in the synthesis have lower purity level than the drug substance, and the components can affect the purity of or may react with the other chemicals during the synthesis of drug substance. The impurities present in the solvents used in the synthesis and the intermediates formed during the synthesis are also generally of less impurity as compared to the drug substance, hence can form impurity in the final drug product. There could be formulation related impurities. The excipients used to formulate drug product can originate potential impurities. During formulation, the drug is subjected to a variety of conditions like heat, shear, etc. that can trigger undesirable reactions. Hydrolysis takes place in solutions and suspensions that leads to degradation. Residual water in dosage forms of solid state such as capsules and tablets provides ripe situation for hydrolysis and metal catalysis. Oxidation occurs for easily oxidizable materials if no precautions are taken. Phytochemical reactions are feasible for light-sensitive materials. Lyophilization and vortex mixing, if used during process of formulation, is considered high-risk operations leading to impurity formation. The other source of impurity could be degradation related. The impurities generated during degradation of the API and/or other interactions on storage. Therefore, it is important to conduct stability studies to predict, evaluate, and ensure drug product safety. Stability studies include evaluation of API stability, pre-formulation studies to evaluate compatibility of API with excipients, evaluate its stability in the formulation matrix, accelerated stability testing of the drug product, and aid in determination of expiration date. The other source of impurity could be due to uh, crystallization related impurities. Some drug substances exhibit polymorphism. The nature of crystal structure can influence the conductivity, crystal shape, color, density, dissolution rate, hygroscopicity, melting and sublimation property, rate of reaction, solubility, refractive index, surface tension, and viscosity. Stereochemistry related impurities. The ICH excludes stereochemical impurities. Pharmacopoeias consider them as ordinary impurity. So pharmacopoeia does not consider them as impurities, and ICH also excludes stereochemical impurities. However, they have different pharmacological or toxicological profile. So it suggests that they have to be monitored very carefully. Genotoxic impurities, compounds, genotoxic compounds can be carcinogenic to humans due to the ability to include chromosomal rearrangement and or genetic mutations. The ICH M7 guideline provide the limits for control of genotoxic impurities in pharmaceuticals to limit the risk of carcinogenicity. So now, as we have already discussed, that all the impurities, any unknown impurity beyond a certain limit based on the maximum daily dose of the drug product, they need to be either identified or qualified. So, what do we understand by identification? The identification of structure, elucidation of impurities, and the drug products is done by either conventional approach or hyphenated approach. In conventional approach, they are separated either by HPLC, ultra high performance liquid chromatography, TLC, HPTLC, HILIC, GC, CE, and SFC. And then finally, the HPLC itself has a UV or a PDA detector attached to it that actually will help in identification of impurities at different RRT. Now, these impurities at different RRT are then characterized for their mass and their chemical structure. Hyphenated methods is a detailed mass spectroscopy which helps to determine the molecular weight of the impurity. NMR and FTIR, they help to determine the various carbon atoms, hydrogen atoms, the position of the various carbon and hydrogen atoms, and also the functional groups that are present, that helps to finally elucidate the final structure of the molecule, impurity. So now HPLC or UV or any other method which is used for determining the purity or assay of a compound, they need to be stability indicating. Why? Because in order to assess or predict the probable impurities in the drug product, they need to be as per guidelines also, and as such also they need to be subjected to stress testing or accelerated stability study and long-term stability study. Accelerated stability study for 6 months will help in when we submit this data to the in the dossier, that helps to obtain a 2-year shelf life. Obviously there are certain limitations to it. They need to be supplemented with the real-time stability data subsequently in order to get a approval for a shelf life. A commitment has to be made to the regulatory agency to submit the long-term data at regular intervals. So now stability, what is the stability indicating method? Definition: validated quantitative analytical methods that can detect the changes with time in physical, chemical, and or microbiological properties of the drug substance and the drug product that is specific, so that the contents of the active ingredient, drug product, and other components of interest can be accurately measured without interference. Very important. They need to be accurately measured, isolated, without any interference. So these are stability indicating methods. The various steps involved are: step 1, critical study of the drug structure to determine and the likely decomposition route. Step 2, collection of necessary information on physicochemical properties of the drug. Step 3, stress decomposition or degradation studies. Step 4, preliminary separation studies on stressed or degraded samples. Step 5, final method optimization and development. Step 6, identification and characterization of drug products and the preparation of standards. Validation of stability indicating assay methods. This is a flow chart of how a degradation force degradation study is carried out on the drug substance and the drug product. If we see the drug substance, solid or solution suspension, photolytic degradation study, thermal degradation study, or accelerated, that means both thermal and humidity stress testing condition needs to be provided to the drug substance, and the degradant that are formed needs to be identified. Solution and suspension of drug substance can be tested for acid-base hydrolysis, oxidative hydrolysis. The details are also elaborated in the guidelines. Drug product, whether it is solid, semi-solid, or solution. For a drug product which is a solid dosage form like tablet, capsules, soft gels, needs to be tested for photolytic, thermal and humidity, and oxidative degradation. If it is a semi-solid, that means it's ointments or pastes, they need to be tested for photolytic, thermal and humidity, that is accelerated temperature and along with the accelerated humidity conditions. If the drug product is a solution or suspension, that is if they are a syrup, they need to be tested for their photolytic, thermal and oxidative degradation. So now a brief overview of quality by design and design of experiments, how the method needs to be developed for stability indicating methods or any impurity isolation methods. Quality by design, the assessment of the purity, related substances, all needs to be all need to be designed. The protocol for analytical validation needs to be designed. The quality needs to be built in the protocol itself. So there are certain approaches which needs to be undertaken so as to build in the robust analytical method validation method. Uh, Quality by design and design of experiments is nothing but it is defined as a systemic approach to the development that begins with predefined objectives and emphasizes on product and process understanding and process control based on sound science and quality risk management. The first step is analytical target profile that needs to be identified. All needs to be listed. That is, it describes the method requirements expected to be measured. What are the uh, categorical as well as the numerical factors that needs to be assessed and measured for a robust method. The design of method itself, feasibility and or availability of instruments are checked. Experimental design is prepared based on the raw materials, availability of raw materials, compatibility. The method is developed. Third is critical quality attributes. The possible effect of factors on the method development is studied. Any numeric or categorical factor, if they are varied, what is the impact on the output of the method? The fourth is risk assessment. It provides a link between the input process variables, numeric or categorical, and critical quality attributes. What is design of experiment? Optimization by response surface methodology is done by numeric variables to estimate the interaction and or quadratic effects. So the various, for example, the flow rate, uh, that is a numeric factor that needs to, and also the diluent ratio, whether gradient or isocratic, not isocratic gradient that needs to be optimized by. They are the various factors or input variables and the output depending upon the various responses that are received. They are the output variables. So design of experiment is nothing but their various input factors uh, are optimized at there are various designs available. They are optimized. Trials are carried out at fixed levels, uh, and the results uh, are then analyzed further. Data analysis is done to obtain a design space. Method verification, the chromatographic space evaluated by the DOE is validated usually in accordance with ICH Q2R1 guidelines. And then there is a control strategy to confirm that the set method performs consistently and as intended to give accurate results. Finally, the life cycle approach, continuous improvement of the method performance based on the design space that allows flexibility for continuous improvement of analytical method without a prior regulatory approval. So thank you. That was a brief overview of how method development, analytical method development is done, why is at all it is needed, and to what purpose, and finally the specification of drug substance and the drug product needs to meet the standards, ICH standards for impurities. Thank you. And ultimately, we have to realize that these characterization, these reporting, identification, and qualification thresholds ultimately impact the safety and efficacy profile of the drug product. So that is why these guidelines are very important to determine the. It's important to determine the impurities that are arising from the drug substance or arising during manufacture or stability or stress testing of the drug product and the residual solvents, which are also regarded as impurities. Not only this, now even the metal impurities, heavy metal impurities, nitrosamine impurities, all these impurities, many of the impurities with the potent genotoxic or carcinogenic potential needs to be identified and qualified. Thank you.